So, chapter 17 of the OpenStax biology book goes over our immune system and disease. And the first section starts off by talking about viruses. So, viruses are a hot topic. It's something that comes in and out of the news and tends to get people's attention when they're hitting. Honestly, I think one of the best couple of paragraphs that I've read in the OpenStax book is actually this first one, where it talks about viruses. And I want to highlight some of the things that they said here. They basically said, we don't know where viruses have come from, their evolutionary roots remain unknown. It's important to know that viruses are acellular parasitic entities and they're not classified in any domain and they're not considered alive. Here they talk about how they have no plasma membrane, no internal organs, no metabolic processes, and they don't divide. They do replicate, but they're entirely dependent on their host cells, and they do not metabolize or grow at all. So they're assembled in their mature form. So viruses are not considered living, and if you wanted to know if something was a virus or a bacteria, basically you could see if it could metabolize, grow, and replicate, and if it could, then it's probably a bacteria. So, viruses, how are they first discovered then if they're so small? Actually, they were first discovered by a filter that was used to eliminate bacteria, and it would still allow for smaller infectious particles to pass through. And so basically, that led researchers to look further and look further until they found out what this was. So, virions are these small particles, and they are just very hard to see. I actually have this cool thing from the University of Utah that I like to show that shows you how small a virus are or is. And they are if you were to look at something like a coffee bean, and this would actually be a huge coffee bean as far as how big my screen is right now, and this is a grain of rice and a sesame seed and a piece of salt, and down here would be, I think, a paramecium, and way down here above the paramecium, if you have really good eyes, you could see the size of a human egg. We actually have really big cells. Are one of our biggest cells is actually the human egg. So as I go in and in further and further, there's an amoeba, a paramecium, there's a human egg if I were to scroll in on it, and then further photoreceptors that help us see color, and a sperm, much smaller than the egg. We've got a single yeast and an X chromosome, and bacteria and mitochondria. Here's bacteria. And as we zoom in further, you can see the measles virus, the flu virus. You can see HIV, a phage that attacks, it's a virus that attacks a bacteria. Hepatitis, the rhinovirus, you can see these get very small. And then antibodies and hemoglobin. And we will start in parts of our nucleic acids, and as I zoom out you can see how small that is. So that's on University of Utah's website, and it's called their size and scale, and I really like looking at it. So the way that viruses are classified has changed over time. Currently they are classified by how they replicate. So a virion consists of a nucleic acid, an outer protein coat, and sometimes an outer envelope made of protein or a phospholipid that the virus derives from the cell that it is on. And you can't actually assume that just that viruses will be more complicated if they are infecting a more complicated being. Actually, if you look back at that from University of Utah, if you look back all the way to these, viruses, the influenza virus, HIV, they don't look that complicated. This phage here that attacks bacteria, that's pretty complicated. And so it's interesting. Their complexity isn't related to our complexity or how we would view or expect them to be more complicated. So virions have a nucleic acid genome that's covered by a layer of protein called a capsid, and the capsid's subunits are called capsomeres. And then viruses also have a viral envelope that has glycoproteins on it that help it attach to whatever cell it is going to be infecting. And then the surfaces of many cells have receptors for, various functions. So the surfaces of regular cells will have receptors on the outside for different functions that it has, and the viruses will come and attach directly to those surfaces. So viruses use the surface receptors of the cell to attack them, but then viruses must have a viral receptor that matches it, or it won't be able to fit. And the host cell that it is attacking needs to be making the types of things like enzymes that the virus needs to survive, or it's not going to be a match. So certain viruses are I don't know if I want to say stronger, but they will last longer. There's two general types that we see, and one is ones that have an envelope around them, and then those that don't. The ones that have an envelope, the envelope makes it a little bit more fragile, so it'll consist of a nucleic acid like DNA or RNA. It'll have capsid proteins and then a phospholipid bilayer around it. But the envelope is kind of fragile on viruses, and it's not as resistant to change in pH or temperature or with disinfectants. But viruses that have no envelope, they tend to be a little bit more resistant to those changes because it's not as fragile. So here's just some more viruses for you to see here and what they look like. It's some of the same that were on University of Utah's site. Okay. So this is a little bit ahead of where we need to be, but picture-wise, but we'll talk about a few more things. So viruses use genetic material. They're just bits of genetic material that are left over, nucleic acids. They're just these tiny bits of nucleic acids, and it can be DNA or RNA. Some that are use DNA bits are chickenpox, hepatitis B, genital warts. Some that use RNA are hepatitis C, measles, and rabies. And how they take over or infect another cell is that they follow a certain cycle. The cycle is going to be an attachment, and here I'm going to be walking through these. They will have attachment, a penetration where they get inside, they uncoat and break themselves up, they replicate, they assemble, and then they release themselves out of the host cell. So when a virus infects the cell, it can cause changes to this cell. It takes over this whole cell. It doesn't care what it does to that cell. And some cell in these changes, they're called cytopathic changes. Some of these actually cause the cell to die, which is called apoptosis, where the cell actually has a programmed suicide. That programmed suicide breaks open the cell and releases all of the new virions that have been replicated. Another way that the cells can do this is the viruses will actually use budding and will one by one bud out and release the new virions. So this is somewhat similar, and when we get to bacteria, bacteria and viruses are similar because they both have DNA and RNA, and they can be harmful to their host, and they can both be vaccinated against. The difference is that bacteria are a living thing and viruses are not. They are just some genetic debris and junk that is left over and can make us sick. Um, so uh, let's move on to, let's go down. How can we treat these guys? So how can we treat viruses? Your own body can sometimes process these and beat them or not. And there's also vaccines and antiviral drugs. We'll talk about your immune defense in the next section, but for here, we're going to talk about how vaccines work. They work by prompting your immune system to develop its own natural defenses. So a vaccine can be prepared by using a weakened live virus or a dead virus, or just using some of the subunits of the virus. So just using little bits of the virus. And this is injected into you, and having the weakened or killed virus or little pieces of the virus that will cause a response basically get your body to respond to it. And then your body knows how to defeat it when a complete virus enters your body. So it's kind of, I kind of think of it as like on sports, and on a sports team, if you were able to get the other team's playbook and you know exactly how they're going to run all of their plays, then it's really helpful in knowing how to defeat them. So one weakened live virus that has been defeated is polio, that they were able to mostly eradicate through using a weakened live virus. So how is a weakened live virus formed? I mean, that seems like it would be terrifying or something. Well, for the most part, it's not. What happens is the virus is actually grown in a weakened, it's called an attenuated form of the virus is built, and they weaken the virus by exposing it to non-ideal temperatures or non-ideal pH, so that the virus is not in its best ideal setting, and so the virus is not as strong as it would otherwise be. And in that weakened form, then it is injected into a body for the body to see what this virus looks like, to defeat it really rapidly, and then develop long-term memory on how to defeat that virus should it come up in the future. Now, the big scare for people is always like, could this thing, mutate and grow into something more powerful? It could, but it's extremely rare. One case is mentioned in your book, but generally it is fairly rare. There are some vaccines that have to be developed constantly. One of those is the flu, one of them is HIV, and that's because the virus mutates so quickly that the vaccines have to be in constant development. And then there's other vaccines like measles, mumps, rubella that have had the same vaccines for a few generations because the virus doesn't replicate, or I'm sorry, mutate very quickly. It's also important to mention that every year around flu season, cold and flu season, people will ask for antibiotics from their doctor to fight what the doctor knows is a viral infection. Well, antibiotics kill bacteria, not viruses. So if you're sick from a virus, an antibiotic won't help you. And in addition, it could actually hurt you because you have a lot of good bacteria in your body that helps you. And in a lot of ways, we're kind of over medicating and killing the good bacteria that we should have a good symbiotic relationship with. So um, there's some vaccines and antiviral drugs that can be given to stop a virus before it really takes over. So for one example, there's rabies. Rabies takes about 2 weeks to really take off and do a lot of damage to your body. So if you develop rabies, say you get bitten by something, an infected animal, then you have about 2 weeks to go and get a vaccine, and the sooner the better. But the sooner you can do that, the better, and you can actually stop rabies from taking over. Another one is Ebola, that has a 70 to 90% death rate. 70 to 90% of the people die from Ebola that contract it. But again, if you get a vaccine for it, then within 2 weeks of getting it, then that can increase your chances of living greatly. So there's different, the way that antiviral drugs work is they usually block the proteins that are in the virus. So usually it blocks the virus from either replicating, or it blocks the virus from being able to attach to a cell in the first place. So it stops those things. Um, so antiviral drugs that they've developed are for like genital herpes or the flu, like Tamiflu. Then there's ones like anti-HIV drugs. The HIV actually, it has a really complicated replication cycle where it has many steps before it can be fully replicated. And so doctors and scientists will develop a lot of drugs that stop it at various points in its replication cycle. In those cases, people will take a whole handful of pills called a drug cocktail, and that's because those meds are trying to stop the virus from replicating at many different points, so that as the HIV mutates, at least something is probably sticking along the way.